morning, everyone. I believe everyone here is, expert from all the local and also international universities here. So thanks again for joining Apical Scientific organized on this eDNA and recon sequencing webinar. So I hope today's this sharing will actually get a fruitful information to and also some insight information for your later research or your upcoming research. All right. So today here, actually we are talking about the topic of environmental DNA. So how we actually use the NGS method to actually attain the study of environmental DNA. So this is another advanced version from what we have commonly known for 16S, ITS, right? So not limited to only 16S and ITS. So we advance further to other gene of selection. So before we start, we wish to introduce the speaker of today, who is Dr. Ahmad-Kamal Ghazali. He is experienced and also expert in metagenomic studies. So I will pass the floor to Dr. Kamal to share with you for the following webinar information. Welcome, Dr. Kamal.
Right. Thank you, Dennis. Assalamualaikum and good morning to everyone. And thank you for the introduction. Welcome to our NGS webinar. Our title for webinar today is from eDNA NGS Shield, Unlocking the Ecosystem Secret, Advanced and Application of Environmental DNA Technology. Right. Please stay until the end of the session because at the end of the session we have a quiz and there's a gift to be won. So you need to stay focused in my presentation because all the answer can be found in my presentation. Right? I will explain to you the price later, so it will be very nice, something that is unique, limited edition. So stay tuned at the end of the session. So okay. Okay, if you have any question related to this, our service, eDNA or amplicon sequences or anything related to what I'm presenting today, you can type your question in the chat box. There's a chat box on this MS Team window. So please write your question and do not forget to write in your name and your email. We'll try to answer this question at the end of the session during the QA session, and those who we are unable to answer during the session, we will answer. We'll be right back to you by email after this. After we have this webinar session, right? So okay, everyone's ready. So let's start with our webinar today. Right. So as a presentation overview, so we'll have introduction to DNA. So we're talking about what is DNA and what is eDNA barcoding, and then some information regarding the comparison between the tools about the eDNA metabarcoding, and then we move on into the eDNA metabarcoding workflow. What are the steps for the eDNA metabarcoding? And then I'll talk a bit about our service and bioinformatic analysis. And finally, we'll go into the demo report. How is our result looks like when we deliver a meta barcoding result to our customer. All right. Okay, so let's start with the introduction. So what is a eDNA, or a short form for environmental DNA? So eDNA is a genetic material that organisms leave behind in the environment, such as in soil, water, or air. So this DNA originated from various sources, including from their skin cells, their faces, hair, or even during the even from the microbial activity, the degradation of the cells. Right. In the environment like soil, water, air, where all these organisms, they are organisms as small as a bacteria or fungi, or up to as large as a large whale in the sea ocean. So these organisms play important roles to ensure the balance within the ecosystem. So this organism constantly release the DNA into the environment, either to organismal shading, for example, like skin cell, because skins always been replaced with new ones, skin cell dead and then replaced by new one. So these cells will be released to the environment. Right. When the animal dies, also, so the animal will be degraded, will be reduced, and then the DNA will be the cells, the tissue, and also the DNA will be released to the environment. So by applying molecular biotechnology tools, for example, like PCR or DNA extraction, we can capture this eDNA, and subsequently we can study the organism presence in the environment, or in different words that we call it as a eDNA barcoding, right? So what is eDNA barcoding? So eDNA barcoding is a method used to identify species based on a short standardized region of a genetic material. So when it means like a short standardized region or genetic material, so for example, like we're talking about like CTNS, ITS, something that is constantly present across the environment. So we're looking at this short standardized region. So we use this genetic material so to study what are the species, we map back to the species that is present in the environment. So so this is the source is often collected from the environment samples, like from the soil, water, or even air, right? So yeah, so the question is what we can use this eDNA barcoding for. So there are a lot of application, most related to like ecology, monitoring. For example, like we would like to see the restoration. For example, there's the land has been you need to, let's say, restore back due to, for example, the fire has been disrupt the wildlife over there. So you want to recover back the forest. So you want to see how the restoration looks like. So you want to study whether the animals, the insect, or the plant there are they being recovered properly. So we can use eDNA to monitor them. Right. We can also look into the monitoring the threatened taxa. For example, if there's a fish that uh, we call it as a something that because of perhaps because of human activities causing the depletion of this species. So we will know the presence of this organism. We can study the abundance of the organism. Are we able to maintain the numbers of these threatened taxa? So we can do something like studies or something like to monitor to ensure that this animal will not be extinct. So that is something we can look for. Or another way is to look into detection of invasive species. But like currently in Malaysia, we have this issue of a catfish, right? So we know that catfish has been invade in most of our Malaysian rivers, so and they are widely disrupt our fish environment. So this is a method actually uh, useful to study not only the presence but also the abundance of this invasive species. So like for example, if there's a fisheries people around here, so perhaps this is some method which is very useful for you to monitor this invasive species. If you know that the number of invasive species is high, then you know that you need to do something on the environment, whether you need to capture and remove all this fish from the environment. So this is some example that you can use for the eDNA barcoding. Right. So eDNA relative is quite it's been established somewhere around year 2000. So previously, before we have this eDNA approach, the method is always been like been captured and released, right? So it's time consuming. It's you need to spend a lot of cost because you need to, for example, you want to study about the fish that in that spot of ocean. So you have to rent a boat, go there for a few days. You have to capture the fish, and you have to release. Or sometimes you might need to capture, and then the fish might die. So that's what we call it an invasive method. So it's very time consuming, and it's a very expensive. Not to mention, it take a long time to complete the process of monitoring this the species and the environment. Um, yeah, so it's not easy on the traditional method. So having a DNA is simplified the process, because eDNA is a taxa specific. It depends on the marker, the gene that you use. For example, if you want to study only a fish, right? Because let's say you're talking about the ocean, so there are a lot of organisms like crustacean, fish, jellyfish. So let's say you want to just focus on fish, you can just use a specific gene marker like COI, which has been widely established for fish metabarcoding. So you can use this gene marker to study monitor the abundance of the fish around. Not only the abundance, but also the identification of fish in that surrounding area. So yeah, so it's easy, it's cheap. If compared to the traditional method, it's easy, fast, less time consuming, and it's very efficient. So that is why we can see in the publication more and more has switched from traditional method to a new method, which is the eDNA method. Right. So talking about the eDNA barcoding, so there are a few ways to actually study this eDNA barcoding. So I can split it into two types, which is the sequencing approach and also the non-sequencing approach, right? So for sequencing approach, we have amplicon sequence analysis, right? That's the first approach. Second, we can look into you can use a metagenomic sequencing. The third will be the DNA barcoding using a Sanger sequencing. There will be a small scale approach, right? Right. And for non-sequencing approach, we have a qPCR and also a hybridization-based method. So technically, there are these a few methods that we can use to study the eDNA barcoding. So let's look into the comparison between this approach, right? So for all this approach, so amplicon sequence analysis, so we can see that for application overview, the application is very specific. We are looking to a specific text of a specific gene marker, for example, like looking at COI, ITS, to name a few. Right, so it's very specific. So if you're looking for fish, then you will catch only fish, COI. So that's why the specificity is very high. Text size is also very specific. So we only capture a type of group like fish. You won't get if you use a fish COI, so most likely you won't able to find like crustacean in your sample. The cost is intermediate, all right. Then let's move on to the next one is the metagenomic sequencing. So for metagenomic sequencing, we collect the eDNA, and then from the eDNA we can do a direct sequencing. So using this approach, we just sequence everything inside the DNA sample. We don't really specify any taxa. So in this case, we capture anything that's inside the sample. So whether it's fish, crustacean, or even any animal that can be found in the eDNA. So the taxa is non-specific and cover wide taxonomy, right? Sorry. However, because we have to sequence the samples, we have to sequence kind of huge amount deep coverage. So there's a cost for it. So normally for metagenomic sequencing, the prices won't come very cheap. So we have to spend a lot just for sequencing, and also the bioinformatic analysis also tend to be quite expensive. So the DNA barcoding quite similar to the amplicon sequencing, except that this is more for a small scale. So you might to focus on a few samples. So DNA barcoding can be used. It's similar to amplicon sequence analysis. The specificity and TASA is similar, except that since that you're working with a small number of samples, the cost related to be quite simple. Yes, we move on to the next approach. Right. So next is the qPCR approach. This is useful if you want to quantify specific DNA sequences, want to study the amount, the number of the species in the environment. So the qPCR will be quite specific, quite accurate, right? But this is will be a non-sequencing based approach. So you need to know the information beforehand before you start with this the analysis. So you need to design the primer. So you need to know that whether this primer used for qPCR will capture the target organism you're looking at, right? So since this is a small scale of analysis, the cost also relative quite small. So this is same goes for the hybridization based method, except that this is instead of using qPCR, this method use a hybridization based method like probes. So this is quite straightforward, easy. For example, will be if you go for field sample, the hybridization will be quite easy to use. All right. So same goes for this approach, same like as the qPCR. The specificity and data size very, very high, but and the cost also relative quite low due to the number of sample that can be used for the studies. Right. Let's move on to the limitation and also the application for this eDNA barcoding. So for limitation or application for amplicon sequence analysis, uh, your the primer might miss the non-target species like I mentioned earlier. So if you're looking for fish, so you might end up missing other organism like crustacean and other organisms inside the seawater. So and also there might has a potential PCR bias there. So because you have to PCR your sample, so PCR bias might be introduced if you're not carefully run the PCR during the PCR process. Right. For application, amplicon sequences is useful for if you want to look for if you want to do a biodiversity monitoring and conservation assessment because it's very wide. So it's not limited to small number of organisms. So this is a very good approach for biodiversity monitoring and conservation assessment. For metagenomic sequencing, uh, the cost of running the metagenomic sequencing is relatively quite high because running the sequencing, you need to have a very deep coverage of sequencing data, and not to mention running the analysis also you need to have a computational power. So that's come with a cost. And sending the samples for metagenomic sequencing, unlike amplicon sequence analysis, since that you only need to PCR, so you don't need to have a very high amount DNA sample. But for metagenomic sequencing, you need to have a high amount of DNA, and also the quantity and quality also must be achieved to ensure the success of DNA sequencing, the metagenomic sequencing. Right, so that's a limitation. So you need to have a high amount of DNA, especially if your source of samples is low abundant of organism, or perhaps they have a high amount of contaminant. For example, in soil, you might have like a phenolic compound that might cause a problem on the, let's say, during the library process. So this might contribute to issue of difficulties of running a metagenomic sequencing, right? For this, the metagenomic sequencing is useful if you are looking for microbiome profiling, or you want to look into the functional gene analysis, species discovery, or comparative genomic. So this method is useful to look into this application, right? DNA barcoding, so similar to the amplicon sequencing, except that this is only for small scale, right? So it's good if you are looking for small scale, but if you go for large scale analysis, DNA barcoding is not that kind of like useful in this case. So you can use that for biodiversity assessment and also ecological studies. For qPCR quantitative, it's limited to the predefined target, less information on community structure. So you need to know the species that you're working on. Otherwise, you might miss on the target you're looking at. This qPCR method is useful for monitoring species, specific species, and also abundance estimation. For hybridization-based method, you may not be able to detect novel sequence, so you need to have information prior of having this hybridization kit based. Otherwise, you might miss your target organism. So required prior knowledge of target is very important. This is useful for targeted species detection and also ecology assessment. Right. Okay, so this is a comparison of eDNA barcoding method comparison. But for our today's webinar, I was just wanting to focus on one method, which is the amplicon sequence analysis approach here. So this is something that is we have well established, and we like to share our experience on having running the eDNA barcoding, meta barcoding using an amplicon sequence analysis approach. All right. Okay, so yeah, so let's move on to the eDNA metabarcoding amplicon sequence using an amplicon sequence analysis approach. All right, so okay, so basically for the eDNA metabarcoding workflow, I can split it into three phases or three steps. Okay, so starting from the sample preparation. Okay, so starting with the environmental bug assessment collection. Let's say if you are collecting water samples, so you need to sieve using a suitable filter size, right? Or if you're collecting from soil, so you need to collect the soil and also how sieve, remove all the unwanted stuff, right? So we need to collect the samples, and then once returned to the lab, so you need to do the DNA extraction, right? So that's a part of a DNA extraction. All right. So once we have the sample prepared, then you can move on to the second stage, second step, which is the PCR, library construction and sequencing. So during the steps, you can start to PCR the sample using the target region. So you need to define your target region. And once you PCR the samples, then you can start to use this amplified amplified DNA for library construction. So for example, if you use a MySex, then you can ligate to the Illumina adapters. So once the library constructed, then you can start doing the sequencing. And once the sequencing has completed, you will generate a lot of sequence data, and then you can use the sequence data to uh, the multiplex construct consensus, or what do you call this, something like amplicon sequence table, right? So in this table, you can match it this to a specific database, and you can do the taxonomy identification. So from the outcome from this, you can identify what are the species that might be present inside the environment, as well as you will get the abundance information. Right. So I will go in deep on this of the few steps. The steps one, the sample preparation, sample two, the PCR library construction, also the bioinformatic analysis, to discuss or to share what are the importance that that you need to have a good result of the eDNA metabarcoding. Right. Right, so let's start with the first step, which is sample preparation. So this is a very critical step, a very crucial step when running the environmental DNA. If you are unprepared or if you did not capture correctly, so it will affect the result quality and also the reliability. So there are a few considerations, for example, like the methodology on the sample collection. For example, you go to the seawater, you want to collect the sample, so there are filtration that is needed, right? So when you filter the water, what are type of filtration paper that you use? So are this filter water able to capture the organism that of your interest? Okay, and sample depth also very important. You know that the water, they have a different water depth of water column. So the upper one might have a different type of organism compared to the bottom. So you need to know what's your objective. So if you capture wrongly, then your intended organism will be missed in your result. This is same goes for the soil. So the soil, they have a different depth, they have a different organism. So you need to know, you need to have a understanding of before running the sample collection, right? So that the result will be as what you intended to be. Okay, and second is preserving the collected samples. So this is also very important because when you're talking about the eDNA, you have to go to the field sample. So sometimes it's not like a day trip return. So it might take a few days. So you might have to think about how to preserve your samples. For example, if you collect the seawater, so you might put it in the large tank, and then you have to preserve in the dark, what they call in the dark room, so that you preserve not only the dark but also temperature must be constant so that you preserve the DNA inside the sample. Or the best is you can filter the water, right? You filter out the water, then you can keep the filtered paper in the perhaps in the cold storage until and then transfer back the sample to the lab. Okay, this is if you have a problem on transporting the samples to the lab. So this is a delay process sample. So you have to consider on this how to preserve the sample once you collect from the environment. Okay. Uh, Another things that need to be considered also is a sample replicates. Okay, so sample replicate is a very crucial, but it's always neglected by customer because we understand that having more samples, having more replicates, increase the cost of your project. But then having replicates is increase the confidence of the result generated. Okay, so having something that is consistent, present across your replicates, it gives you confidence that this result is true, rather than having one samples and then you have one organism, only one samples, right? You don't have any replicates, and then you might found that this species is high, but you don't know, perhaps because of issue with your sampling, or perhaps something related to your methods on collecting the sample tends to have some issue in the result. So that is why having a replicates will tend to uh, control these biases. So having a replicate is very important, is very crucial. We advise that to have a minimum of 3 replicates per group. It depends on the environment, I can say, but for us, Apical Scientific, we request that at least three replicates per group for us to allow having a statistic analysis. When I mean the 3 replicates per group means that let's say you want to do a comparison analysis from seawater at a different time point. Let's say you want to collect samples at January, and then you have a second group which is at June, for example, early and the middle of the year. So at January you need to have three replicates, three samples, and also at a June time point also you need to have another three replicates. So in other words, that for these 2 time point you need to have three replicates, in total 6 samples. So that is mean by 3 replicates per group, right? For us to have a statistic analysis, and this is very important if you want to have a good confidence on the result. Okay, the more the better, right? But minimum 3 replicates per group is needed for statistic analysis. Right. So for sample preparation, if you are sending the sample to us, so as a DNA, let's say if you extracted the DNA, so what we need is a genomic DNA, which is amount of 200 minimum of 200 nanogram per microliter in volume of 20 microliter. So the concentration must be more than 10 nanogram per microliter, and the purity must be zero. Purity of OD260-O must be 1.8 to 2.0, no gradation or no contamination. If you have difficulties on extracting the samples, for example, you hardly to achieve our requirement here. So we do offer DNA extraction service. Do contact us for further information on this. So we have established a lot of experience on how extracting DNA from customers' eDNA samples. So our method is something that it can be rely on. Right. So second step is the PCR. So once you have a DNA extracted, so next is important is to look into the PCR library construction and sequencing. So for the PCR process, as I mentioned at the very early stage of this EA webinar, the gene target is important. So it covers a specific organism group. For example, let's say a 12S, you have you can use it to capture fish, right? And then you have a cytochrome B, COI, 18S. All these are very specific target group, right? So it's very important your intended group in of what you intended to capture. So my advice is if you want to know what are the gene target that you should use for your project, the best is try to look into your publication that might relate closely relate to your project. That would be the best advice I can give. Which can try to use their approach, use their target region to use it applied on your study. So that would be the best approach advice I can give to you. Not only that, not only about the gene target, but you also need to look into the database availability. Uh, this is a very important consideration because unlike, let's say, for bacteria apical sequence analysis, we can always suggest that go for 16S. And for 16S, we have the database has been well curated, well established, like the Silva database is well established. But unlike the bacteria, so like the fish, eukaryote, and all these eDNA metabarcoding, the database is kindly like still been built up, right? So you need to look into you need to check what are the database that is suitable for your studies. The best also the same advice as I give earlier is to really go back to the paper, see what are these paper this author used to match the sequence back to the sequence. What are the database that used to do the species identification? Like for example, like insect, you have the coins database, which is quite a good database, quite good established database. And for fish, I think you have a few database. Sorry, something that has missed out from my mind. So, but there's a few database for fish COI, and this is quite curated and specific COI database created intent for the specific target organism. For NCBI database, I know some customer would like to match to the NCBI database, but NCBI, as you know, is not that accurate compared to the database that I mentioned earlier, because NCBI consists of a lot of organism, and when you match your COI, it can match to a non target organism, and then your result might not be accurate. So this is something that is not advice. I mean, I would not advise to use an NCBI database to do the matching. And sometimes when you see the taxonomy, what they call the taxonomy lineage for NCBI, this is not standardized compared to like the curated database. So you might your result might be a little bit what they call not well organized in terms of the taxonomy identification. So the best is you try to use need something that specific database which has been well curated for the taxonomy identification processes. Okay. Yeah. And for the primer, there are a few available, so as I mentioned earlier, so you can always go back to paper and see what are they are doing. Like for example, like Illumina, they can use you can use something like this paper sourced by Star et al. So they use this primer. And also you need to consider the sequencing platform that used for the eDNA, right? So for in our company, we use a MySec, so we have in-house MySec to run the sequencing. So MySec can accept amplicon size up to 500 base pair. So for example, like anything size amplicon size up to 420, we can handle the what they call the sequencing of the eDNA. Okay, right. So yeah, always go back to the paper, see what are the primer that use, also the database, what are the database they use to for their study. That will be the best advice that I can give you for eDNA studies. All right. Uh, for the our sequencing process, library PCR, library construction and sequencing process, for our standard process, normally we will do a QC on the sample. So once, let's say, you send your sample to us, we do a QC to check whether the QC does achieve the OD to 60, to CO280 of 1.8 to 2.0, whether we have enough amount of DNA. So we need to check, and we once the QC is passed, then we can do the PCR QC. So we have a PCR QC to check that whether the sample that sent to us can be used for the PCR process. If both sample QC and PCR QC is passed, so we will notify to the customer that the sample is good to be run for the PCR library construction and sequencing process. So we update the customer by email about the QC status. So during this process, during the sequencing process, the outcome from the sequencing process, so we generated more data of 200 raw reads or equals to 100,000 raw text per sample, right? So it depends on your size of the reads, the size of your target region. If let's say your target region is size about 420 base pair, like I shared earlier, so we can use a sequence format of 300 PE per N to capture this 420 base pair target region, right? The outcome will be in the FASTQ format, all right? So this is a general FASTQ raw data generated by the next-gen sequencing machine. Okay, all right. So yeah, so at the end, this step is you have a raw data. So it's just a raw data without much information. So to get most information, so we need to process it. All right. So just a simple workflow. So we have done the sample preparation. So we have collected the samples. I have explained what the next section, and then then move on to the PCR, library construction and sequencing. Right. The outcome will be a short read sequence generated by the sequencing machine like Illumina MiSeq. So you might have like 300 per N data. So it's just a sequence data without any further information. So we need to process it during the bioinformatic analysis. So the bioinformatic analysis will do a sequence clustering. So we'll cluster the sequence to the pipeline to cluster according to the sequence similarity and to call out the abundance. All right. So we have the sequence, the representative sequence of each cluster, and also the abundance. And then using the sequence, the representative sequence, we can match it to the database I mentioned earlier. So from the database, we can identify what are the species are. So we can match the sequence to the species, right? So we have the sequence, and then we have the abundance information together with the species information. So all this information will be available, right, for all the sequence, right? So we have a table providing all this information. All right. So basically, we give a very technically is already good enough for you to have a basic information about what are the species in the environment together with the amount, the abundance, how many they are inside the environment on that particular collection sample collection time. Right. So let's say this is only one sample. All right. So if you have multiple sample, or in this case, a multiple group, as I mentioned earlier, so you intended to compare the samples on different time point on January, June, or January. So you have the let's say this is some samples from January and this is sample from June. So you can process the sample as I explained earlier, and during the bioinformatic analysis, we can combine this result and we can compare it, right? So we can compare the abundance between these two time points. So you know that what happened during these two time points, whether the species are they increased or are they reduced. So it's something that you can look into and study them. Apart from having this table, the information about the sequence, the abundance, and also the species information. There are other things you can do. For example, you can have a statistic analysis, the alpha, beta, diversity, which is a common analysis when you run the meta barcoding. Apart from that, you can do a comparative studies, which for this to be done, you need to have more than two groups, right? So you need to compare. So you need to have at least two groups, because without, if you have one group, definitely you cannot compare, right? So you need to have two groups, and each group we must have two replicates. Okay, so for example, I mentioned just now, so Jan and sorry, from January to June sample. So each sample, sorry, each group you need to have a three replicates. So in total six samples. So you will have you will be able to get the comparative studies result here. All right. Okay. Yeah. So that's what we can what we deliver to you. So if you are sending the sample to us, so I can share with you. We are Apical Scientific. We are have a very wide experience on running amplicon sequence analysis and also eDNA metabarcoding. So we can offer you one-stop solution from sample prep up to the bioinformatic analysis. So we have MySec platform in-house, so we constantly run customer samples. So we have a few runs in one, have a few runs running our MySec platform. So there are few widely used target region, like for example, the COI, TRNL, ITS. This is a common target region used by the customer. Apart from that, we can also accept customer target region, which is up to 500 base pair. Right? If you like to know further about our service, you can contact us. All right? Uh, which I will share you at the end of this slide. Right, before we move on to let's move to the demo report. So we want to show you how our demo report looks like. Okay. Yeah. So just to share you our report of eDNA service. So this is a demo report for ITS. So the result will be more is the same as for other target region. So you have the result will be delivered to you in a HTML file. This is a report, and you have a result where you can open, you can get the sequence, the sequence, the table, and also the figures are in the separate folders. Right. So the report here, the HTML will be combine everything so that you don't have to open multiple folders. So by having this report assist our customer to view the result in one file. Okay, yeah, so we have this project. So you can see there's a tabs here we can you there which represent the analysis that you conducted on the samples. Right, so I will briefly go through the report here. So if you have interest of know further about our demo report, you can email to us and we can send to you this demo report, which you can download for yourself, and then you can have a taste, you can play around with our demo report. All right, so I will briefly go through our demo report. So we have read statistic. We explain about the amount of data starting from the raw data, raw reads generated from the Illumina machine up to the process before running the bioinformatic analysis. And the taxonomy overview. So this is a basic table. So you have the sequence, and then you have the abundance information, as well as the taxonomy information here. So this is some basic information of the amplicon sequence table. So this is a basic data that you can use for species identification and also abundance information. And from here, this data will generate a few type of visualization. For example, we have a Texa bar plot, so you can provide result in the bar plot here. The bar plot is very intuitive, so you can you can see that you move mouse. Then if I move to the sector, then you can see the information will be highlighted around. So you can do a lot of things here. So for example, that you want to focus on certain groups, then you can just go to here, and then you can double click and will highlight your group of interest. Okay, right. And then you have a top 10 result. Okay, so you have a top 10 across samples, all across all taxonomy level like phylum, class, and so on. We have a result in the heat map, also the corona. So we have the result in the corona. So you can please use this data for your publication as well. So there are other type of statistic analysis like distance metric. So we can provide also the UPGMA, and then we have the alpha diversity. So this is what I mentioned earlier in the slide. So we have alpha and beta diversity. This is something that you can use to study the structure of the organism in the in your environment on that particular collection time point. This is on the alpha diversity. You also have ordination, where you can use to compare between groups, right? And then you have a metacoder, which you can also another way of a comparison samples between groups or between sample. You can also do it between samples. So metacoder is used for comparison. And then you have um, like a dissect. So this is another way of you want to compare between a specific group. You want to know what are the species that is highly enriched on the particular group. Then you can use something like comparison, and this is like a dissect. So we can provide you able to find what are the species were enriched or what are the species that might be reduced on the different groups. Right. Yeah, and also you have a Venn diagram. So as usual, so you can compare between groups, so you're able to know what are the species that might be shared or might be unique to respective groups or respective samples, right? So that's basic information about our demo report. If you interested to have this demo report, feel free to email us. Okay, you can email us at this email, and we will communicate to you on the inquiry. Okay, right. So that's last part of my presentation.